So thanks for the introduction, and thanks for the organizer to invite me to speak in this session. So I will try to... so Dan Region has already introduced an atypical way of performing molecular replacement, which is fragment-based molecular replacement. Now, this technique is very different because we are going to use not anymore the full complete model for performing molecular replacement, but we are using partial fragments. So because we are just using a very small part of our structure, then it is required that this part should be very accurate. I mean that the RMST against the final structure should be very low. So attached to this problem, then, it comes out that we need to use a particular approach to the molecular replacement problem, which is based on maximum likelihood, like the one that is implemented in Phaser. And the reason is because this is the natural statistical environment in which we can actually describe the characteristic of these fragments, being partial but very accurate. We also need to rely on data quality. So for data quality, I mean that it is required that there must be, the data should be complete, or as complete as possible. We need to have relatively high resolution data, not lower than 3.5 Armstrong usually. Then we need to rely on information content because we don't want to throw away information from the reflection. We just want to use all the possible information that we get from the data. And whatever combination of molecular replacement software we use, we need, in any case, to extend and complete our structure. This is possible to do by applying density modification and auto-tracing algorithms that will interpret the map that initially we obtained and complete the final structure. And this is possible to do, for example, with ShellXC. Now, on the left, you see a list of software that is currently distributed in CCP4, and they actually work on this area. So all the Archimboldo frameworks, Archimboldo, Borges Shredder, but also Ample, as you already seen, and Fragon. So one of the first things that I want to point out is what actually my program is doing. So what ALEPH is doing. ALEPH is the software that I programmed in Barcelona with Isabel, and is a bioinformatics tool primarily designed to extract fragment libraries from the database. So then this library can be used as search models for molecular replacement. Now, because those libraries because these fragments are very small, we cannot easily rely on sequence identity and sequence alignments. Then we need to have some geometrical descriptors, in particular vectors, and encode the relations among all of these vectors into networks to try to describe the fold. So this is a different approach from the classical ab-initio one, in which we are trying to create de novo the models. Here we are extracting the models from a database, but we cannot rely anymore on the sequence. So because we build up networks, then we can explore these networks. We can apply some algorithms and, for example, understand how a structure can be decomposed in subfolds. And we will see later some example of that. And also, we'll try to use, to provide to the user some sort of superposition algorithm that will help on superpose discontinuous fragments onto a target structure. So we thought that the best way of presenting this software will be together with a graphical user interface that has been programmed by Joseph Trevino in Barcelona and is based on Python 3 and PyQt5. So, as I told before, we need to have a geometrical descriptor to avoid the use of the sequence for extracting the models. And Isabel came already in 2013 with a nice idea of build up some vectors from the main chain. So if you have 3 consecutive residues, you can always compute the centroid of the carbon alpha and the centroid of the oxygen, of the carbonyl, and then you and you just link these 2 points here, and you end up with a vector. The length of this vector is statistically, so the length of this vector is telling us about the secondary structure, because we can, as you can see here, we have 2 different distributions for this length. One referring to the alpha helices, and the other one referring to the beta strand. And then also the direction of the vector is telling us about the secondary structure, because in the helices tend to coincide with the direction of the helix, and in the beta strand we have an alternate conformation. Now, if you provide to ALEPH a PDB, and so all the main chain is explored, and for every 3 consecutive residues, a characteristic vector is computed, and all of these vectors became nodes in a graph network in which the edges are just storing the information about the three-dimensional relationship among them. So distances among vectors and angles. Now, one of the first questions that we need to answer at this point was, what is a secondary structure element? Of course, it might seem a stupid question, because we all know what is a secondary structure element, but the correct answer for us was depend on the use that you want to do with these particular fragments. So if you look on the picture on the left, and if you are far enough, you will see instinctively, in just looking to the picture, you will see that you have 2 helices. But then if you look closer, you will see that there are really strong and major errors on the position of the atoms, and the hydrogen bonds are not actually formed. So we should not classify them as alpha helices. But of course, these 2 fragments cannot be anything else than alpha helices. So if we want to catch both the properties, the local correct property of being that particular residue annotated as belonging to an alpha helix, or if we want to grasp the general description of the fragment, we need to have not anymore a one single definition of what is it, a secondary structure, but a continuous description. And together with Anna Medina, that is a PhD student in Isabel Usson group, we end up building up criteria that we call sensitivity, and this is just a number that is when it's close to 0, it means that we allow the program to be very generous with the annotation, trying to grasp the general features of the fragment. And if it is close to 1, we try to catch the local correctness residue by residue. So this is a parameter that actually can be configured by the user and can to have different annotations. Now, this is an example of what I meant with that. If we start this video, we see this is a structure that has been provided to the GUI, and then you can select 2 different values for the sensitivity parameter, and this is what comes out. So you have some helices, you have some strands, and what is not connected has been annotated as coil. And you can see what is not has been annotated as coil by showing, for example, the template as a trace. So you will see the difference between the full protein and what has been annotated as secondary structure element. Now, if we choose a different threshold for the sensitivity, something that is smaller, then we ask the program to be more generous. And if we go into the other extreme situation, where we are actually asking for a higher sensitivity, then we annotate less. Okay, so as I said before, the focus of this program was actually to create library for molecular replacement, for fragment-based molecular replacement. And this program, ALEPH, is basically using as input a template, which is a PDB file describing the fold, is interpreting this fold in terms of characteristic vectors and networks, and searching this network against a database. The database can can be, the full PDB can be a directory that is provided by the user, can be inferred from a sequence file. And the output will be a directory containing all the superposed, clustered, annotated fragments extracted. Now, the natural way of using this library is together with this program, Archimboldo Borges, in which all the fragments, all the models on the of the library are considered as a whole object. So all of them are explored within Phaser, and we try to grasp the pattern of the rotation and the translation that comes out. Because you have to understand that these are very small fragments, so sometimes only the LLG and the zeta score are not so high enough to discriminate for correct solution. So we need to analyze all of these models and consider all the results all together as a whole. On the left, you will see there are some examples of libraries that are already distributed in CCP4 that you can use, and you can see here that are very ubiquitous, so they can be found in different families. This is one of the reasons why we cannot just rely on sequence. Okay, this is an example of how to generate libraries from the GUI. So you can provide from what database you want to select the fragments, and again, you can do it from the full PDB. You can provide a sequence or even a cat ID to select the fragment. Then, of course, you can select the sensitivity. You can run the program in the local machine or spread the computation on a grid. You can even decide for different algorithm for clustering the models in the library. And of course, there are a set of other settings and parameters that you can configure, and all of them will be documented. Here, I'm just changing the font of the GUI, and this is just for allow me to increase the size of the top of the bottom, in which I can show you an example of the model that has been extracted from using a template. So on the left, it's a simple fragment model that has been extracted from the database, and on the right, you will see the template that is being used. In this directory library, you will have the list of all these fragments that has been extracted. Again, I say this library is ready to work with Archimboldo Borges. Now, I just want to point it out that at the beginning of this talk, I said that because we are using small fragments, then they must be accurate. So there are different ways of achieving this accuracy. One can be to have a large library representing the folds, so you will have lots of possibilities of variation on the same fold. Another possibility will be to work with one distant homolog, one distant model, but trying to break this model in smaller pieces, trying to and then use it, each one of these smaller pieces, these small subfolds, as rigid body groups to performing particular function of rigid body refinement in Phaser that has been already introduced, like gyre and gimbal. So the question now is, how can we discover, how can we decompose a structure into subfolds? So intuitively, if we look to this picture, we would like a structural biologist have these two helices in the same cluster, for example, and these two beta strands here on the same same cluster. But how can we actually do all of this classification automatically? And the way in which I thought to do in my program ALEPH is using an algorithm that is called community clustering, and it's an algorithm that is analyzing the network of all these vectors, of all these characteristic vectors, and trying to find subfolds, so subnetworks of fragments, maximizing the compactness of the fragments. So I can also force the program to trying to get uniform sizes of clusters, and this sometimes can make sense because these function in Phaser, gyre and gimbal, they will work better if we don't have very small fragments on it. And this is an example of a structure 1F0L or OL, and you can see that if we ask the program to create clusters, I forgot to mention that here each color represents a different cluster. So you will see that that if we ask the homogeneity on, so if we want to decompose trying to enforce this uniform cluster sizes, then we'll end up with one kind of classification. But if we switch off this option, then we will end up with smaller but a higher number of clusters. So what is the best classification? Well, it depends on the problem. The good thing is with this GUI and with this program, you can choose, you can see in real time what is the results, and you can try to use one model or another in Phaser and trying to see what works best. So, and again, we can enforce other structural property. For example, it makes sense not breaking a beta sheet in pieces unless this beta sheet is not very large. And we can force the program to do that by applying these other filters, so pack beta sheet on. And you will see, until now, what have we done is decomposing the structure in subcluster, but fixing the number of clusters that we want. Another approach could be to perform this algorithm, but in an iterative hierarchical way. In this way, our question is, what will be the best way of classifying in just one single cluster all the secondary structure elements of my protein? And of course, the answer will be just one cluster containing all the secondary structure elements. But then, if I ask to the program what will be the best way of splitting into the structure by optimizing the compactness, then this is the answer for this case. And I can go on further, trying to splitting in 3 clusters, 4 clusters, etc. And you can see that the result that comes out is a dendrogram. So it's a hierarchical clustering in which that is a mathematical descriptor that actually we can store and use. And you can understand that if we have two structures that are similar, then we expect that also this classification is the way in which they compose. And so the two dendrograms that must be similar. And so this can be a fingerprint, a signature for our structure. Now, the last thing that you can do with this program is superpose small fragments on top of a bigger structure. And this is what is presented here. So you will see that you can load a structure that is, in this case, is the reference, and then you can choose if you want to superpose just one fragment or a directory containing a library containing multiple fragments. And then when you run it, you will see that the first thing that comes out is actually the full target structure. So this is the structure that on which we want to superpose the small fragments. And now we will see an example of how the program. So you will see in blue there are the two helices that are actually a small fragments that has been superposed on this big fragment. And this can be sometime important because classical algorithms can fail on manage with very small disconnected fragment when superposing. So these are my conclusion. I just want to remark that ALEPH is a bioinformatic tool, and it's optimized for user control. Then the way in which we annotate and we interpret secondary structure is not, it's in a continuous fashion by using sensitivity. We can also interpret a structure by decompose in subfolds and use each one of these subfolds as an independent rigid group for Phaser rigid body refinement. And then, of course, the main focus is generating libraries for molecular replacement. And of course, sometimes can be useful to superpose these small fragments to the final structure for post-mortem analysis. So I just would like to thank my current group, Randy Reed group in Cambridge, which I'm working now, to also let me continue working also on this project, and while I was doing, of course, things in Phaser, and Isabel Usson, in which this project is maintained and has been developed primarily, in particular Anna Medina and Joseph Trevino, that are very involved on this project, and also all the collaborators, in particular Josh Sheldry group in Göttingen. So thank you very much.